Assalamu alaikum, everyone. In the next 15 minutes or so, I will present some of the biomedical research by SSC faculty in the areas of drug discovery, antimicrobial resistance, and vaccine design. The biomedical research at SSC is cutting edge, it is interdisciplinary, and it integrates biology, chemistry, computational biology, and engineering to address local as well as global healthcare challenges. We can divide this research into five main areas. The first one is basic biomedical research that includes understanding mechanisms of animal development and control of gene expression. It includes surveillance of pathogen and antimicrobial resistance and cancer biology. The second one is translational biomedical research that includes drug discovery. It includes identification and validation of vaccine targets. It includes understanding mechanisms of various diseases. Then we have epidemiological and demographic research. This includes estimates of incidence and prevalence of disease and identification of risk factors. Then we have computational research that involves disease modeling and outbreak predictions. It includes cancer systems biology. It includes proteomics tool development and genomic analysis. And lastly, we have biomedical devices that includes but is not limited to devices for detection of TB or continuous glucose monitoring system. The modern methods of drug discovery are actually molecular mechanism based drug discovery. So this means it involves identification of a target. A target can be a protein that is deregulated in a particular disease, for example, in cancer or in diabetes. It can be a protein that is required for the reproduction or the survival of a pathogen like a virus or a bacteria. Once a target is identified, it is validated throughout the world in different laboratories. And this validation means you make a similar mutation in that target and see whether it causes a disease state and then whether you can rescue that disease state or not. Once a target is validated, and these targets are usually enzymes, so their function can be inhibited by chemical compounds. You screen a large library of compound and make an assay for that. So this is called assay development and high throughput screening, where you can screen hundreds or thousands or hundreds of thousands or millions of compounds, and you come up with certain hits. These hits are then evaluated in cells. They can be evaluated in vivo, in model organisms. And this is aided by medicinal chemistry, structure-based drug design through a process called lead optimization. And this also involves pharmacokinetics and pharmacodynamic studies. And this process goes around many years, and you come up with one drug that goes through formulation studies in the clinic, as well as toxicology studies, and finally into the clinic. So this whole process, out of this whole process, our work mainly involves assay development, high throughput screening, and mechanistic cell-based studies. So these are the faculty whose work I will talk about today. And these are the 6 areas of drug discovery, out of which we, as I mentioned, we mainly focus on 3 and 4, but we also work a little bit on 1 and 2, and the last one is the one that we don't work on. So the first up is Dr. Shephar Mirza. She works on type 2 diabetes, pneumonia, as well as SARS-CoV-2. Her research can be divided into 2 parts. Number 1 is understanding why the diabetic patients are more prone to pneumococcal infections, and she utilizes various methodologies, including epidemiological and demographic research, immune profiling, as well as computational and bioinformatics-based research. Second part of her research is infections and antimicrobial resistance. Her group is the first one in Pakistan to map antimicrobial resistance. Similarly, she's sequencing pneumococcal strains from Pakistan, as well as identifying why or what sort of response the vaccines produce to the local strains of various infectious agents, and also epitope mapping spike protein of the SARS-CoV-2. Then we have Dr. Shahzadul Hassan. His areas include hepatitis C virus, SARS-CoV-2, HIV, and dengue. He uses various chemically synthesized libraries as well as natural products for his screening studies. He's made various biochemical as well as cell-based assays, and he's identified various molecules that inhibit the entry of the virus. One of them is LUMS1 protein that actually binds to the cells and inhibits the entry of the virus. This protein, for example, you can see when you don't add the protein, the green is the virus that is infecting the cells. And when you add the protein, there's no infection inside the cell. Similarly, he's trying to map neutralizing antibody epitopes from the patients, HCV patients, that recovered from the infection without even knowing that they had the infection. Then we have Dr. Muhammad Saeed from Chemistry and Chemical Engineering. He works on dengue protease, as well as SARS-CoV-2. His work mainly involves assay development, high throughput screening, and lead optimization. So he, for example, takes a target from SARS virus, for example, or dengue virus, for example, and develops an assay and then screens compounds and then tries to improve those compounds. Here are the examples of some of the compounds he has identified. Especially important are these three compounds that he has purified from clove that inhibit the dengue protease. He did this work in collaboration with Dr. Shahzadul Hassan's laboratory. Then we have Dr. Rahman Shahzeb Salim. He focuses on cancer, especially microtubules, protein kinases, and multi-drug resistance within cancer. He also focuses on Alzheimer's disease. His lab works in close collaboration with my lab that I'll show on the next slide. What we are trying to do is that, this is a normal mitotic cell. So when we treat with the drugs, the cells form, malfunction mitosis, and that ultimately leads to their cell death. He's an organic chemist, so he synthesizes many compounds that are based on rational design. They are structure based, they are computer assisted, and we test those compounds in various biochemical and cell based assays. So he characterizes the compound through various chemical analysis. Here are some of the examples of the compounds that his lab has synthesized. My lab actually focuses, as I mentioned, on cancer. We focus on microtubules, oncogenes such as Aurora and FLT3, as well as multi-drug resistance. So the compounds Dr. Rahman synthesizes, we use them in various screening assays. We also use natural products. We've developed various cell-based as well as biochemical assays. And then we also do some cell-based mechanistic studies. This is an example of the compound. So the cells normally have these thread-like structures called microtubules. When we treat with our compound, the microtubules are no longer there. A normal cell divides into two by forming these bipolar spindles. But when we treat with our compound, there is no bipolar spindle formation, and these cells ultimately die. Next, we have recently we were given a grant by Arctic Pharma from Norway to do various cell-based as well as biochemical assays for their lead optimization studies. Next up is Dr. Ghayur Abbas. His diseases and targets involve cancer, bacterial multi-drug resistance, and plant fungal diseases. He's an expert in finding new ways to synthesize compounds. He's especially, for example, he's made these boronic acids and esters that are used in various drug synthesis processes. These are the examples of some of his compounds that he synthesized. And you can also see that he makes this derivatives of this boscalid compound that is a herbicide and there's often a resistance to this herbicide. So he's aiming to produce analogs that can overcome this resistance. Next up, we have Dr. Muhammad Afzal. He works on cancer, cardiovascular diseases, SARS-CoV-2, and HCV. He's mainly interested in making RNA-based vaccines, especially circular RNA vaccines that are more stable. He's an expert in making exosome-based delivery of these RNA vaccines. These are the exosomes that he's isolated here at LUMS. He's established various assays here at LUMS. Next up is Dr. Safiullah Chaudhry. He is a computational biologist. He acquires large biological data. He models them, he simulates them, analyzes them, and then makes predictions. For example, for the personalized therapy, he can take data from cancer, from tumors of the patients, put them in the computer, analyze them, through his software that he's developed, and then make prediction that what sort of treatment should be given. So for that, he has made this theater for in silico oncology. He's also made the first freely available protein search engine. And these are the kind of cells that he makes on the computer. Going back to the original drug discovery process that I showed you in the slide at the beginning, we are mainly focusing on these two areas where we do the assay development and high throughput screening, as well as mechanistic cell-based studies. We can't do in vivo evaluation or clinical evaluation. In vivo evaluation is prerequisite for the clinical evaluation. Similarly, you need pharmacokinetics as well as pharmacodynamics for the compound to progress. So LUMS is currently building an animal house at the back of SSE, and we hope that the mouse facility there will help us in the drug discovery process, especially in the pharmacokinetics and pharmacodynamic studies. And then someday we can take a compound further in the mouse and to the humans. For the infectious diseases, however, we still need very specialized labs. We've got grants, but we don't have those specialized labs yet. So we propose that the right wing of SSC, third floor, be converted into these specialized labs that have the biosafety level 2 labs, that have microbial genomics and multi-drug resistance lab, that has a biochemistry and structural biology lab, that has a biotech innovation lab. We recently received a grant to make the multi-drug resistance lab named after a donor. We are hoping that we'll raise enough money by naming these labs after the donors that can cover the estimated cost of 100 million Pak rupees, and we can continue to do research in these areas where we already got some grants. Thank you for your attention, and I hope that I'll be able to answer any questions you have during the live sessions.